Welcome back to G Fun Facts Online. Today we are diving into the atomic machinery that makes you sentient. In the vast, dark universe of the human body, 1 trillion microscopic lightning storms rage every second. They are the reason you can hear this voice, feel the device in your hand, and think about what I am saying. This electrical storm is not chaotic. It is a symphony of precision orchestrated by a family of proteins that serve as the transistors of the nervous system known as the voltage-gated sodium channels, or VGSCs. For decades, scientists viewed these channels simply as on and off switches, biological pores that open to let sodium ions rush in, triggering the action potential that defines neuronal firing. Today, we know they are evolutionary masterpieces that predate the nervous system itself. They are complex signal processors that can differentiate between a gentle caress and a searing burn, and they are shape-shifting machines that, when broken, lead to some of the most profound disorders known to medicine. To understand how a thought becomes an action, we must zoom in to the scale of angstroms. The voltage-gated sodium channel is a massive transmembrane protein, a leviathan of roughly 2,000 amino acids that weaves through the cell membrane like a suture. The functional core of the channel is the alpha subunit. Imagine a single polypeptide chain folded into 4 homologous domains, labeled 1 through 4. If you were to unfold the protein and lay it flat, it would look like 4 repetitive chapters of the same book. Each of these 4 domains contains 6 transmembrane segments. These segments group together to form 2 distinct functional modules. First, there is the voltage sensing module. This is the channel sensor. The S4 segment is particularly famous among biophysicists. It is studded with positively charged amino acids, specifically arginine or lysine, at every 3rd position. Like a magnet repelled by a like charge, the S4 segment moves outward when the inside of the cell becomes positive, a process called depolarization. This physical movement is the trigger that opens the channel. Then there is the pore-forming module. These segments from all 4 domains come together in the center to form the ion-conducting pore. Between segments S5 and S6 lies the P-loop, which dips into the membrane to form the selectivity filter, the gatekeeper that ensures only sodium passes through. While the alpha subunit can function alone, in the body it is rarely solitary. It is accompanied by 1 or 2 auxiliary beta subunits. These are smaller proteins that look like cell adhesion molecules. They act as chaperones, guiding the alpha subunit to the cell membrane, modulators that fine-tune how fast the channel opens or closes, and anchors that allow the channel to latch onto the extracellular matrix, effectively gluing the channel to specific spots like the node of Ranvier. The life of a sodium channel is defined by a 3-act play that cycles in milliseconds. First is the resting or closed state. The pore is shut, but the voltage sensors are reset, ready to fire. Second is the activated or open state. Upon depolarization, the S4 sensors slide outward, pulling on the intracellular linkers like a lever. This torque opens the activation gate, formed by the crossing of the S6 helices. Sodium ions flood into the cell, driven by both concentration and electrical gradients. This influx is the rising phase of the action potential. Third is the inactivated state. Within a millisecond, the show is over. A hydrophobic cluster of amino acids, known as the IFM motif, located on the intracellular loop connecting domains 3 and 4, swings up and plugs the pore from the inside. This hinged lid mechanism stops the flow of ions, even though the membrane is still depolarized, allowing the neuron to reset. But how does the channel let sodium in but keep potassium out, even though potassium is only slightly larger? The answer lies in quantum-level selectivity called the DEKA motif. The selectivity filter is formed by 4 specific residues, 1 from each domain, consisting of aspartate, glutamate, lysine, and alanine. The lysine is the masterstroke. Its positive charge acts as a bouncer, repelling other positive ions just enough to strip water molecules off the sodium ion. Sodium fits through this stripped-down state perfectly, while potassium is too large to fit without its water shell and too energetically expensive to strip naked. We often think of sodium channels as brain proteins, but their history begins billions of years before the first neuron fired. The ancestor of the VGSC is likely a bacterial channel, similar to the extant knockback. These primitive channels are homotetramers, meaning 4 identical subunits coming together, rather than one long chain of 4 domains. They likely evolved to help single-celled organisms maintain osmotic balance. The leap to complex life involved 2 rounds of gene duplication. First, an ancestral single-domain gene duplicated and fused, creating a channel with 2 domains. Then, another duplication created the 4-domain structure we see today in eukaryotes. This asymmetry allowed each domain to specialize. Recent genomic studies on choanoflagellates, single-celled organisms that are the closest living relatives to animals, revealed a surprise. They possess sodium channel genes. This suggests that the machinery for electrical signaling existed before there were nervous systems to use it. In mammals, there are 9 distinct genes producing proteins named NAV1.1 through NAV1.9. While they share 75% of their DNA, that 25% difference dictates where they live and what they do. We can group them by their sensitivity to tetrodotoxin, or TTX, the deadly poison of the pufferfish. The first group is the TTX-sensitive channels, the classic neuronal specialists. NAV1.1 is the interneuron's metronome. Located in inhibitory interneurons in the brain, it is built for speed. It recovers from inactivation incredibly fast, allowing inhibitory neurons to fire at high frequencies without tiring out. Without NAV1.1, the brain loses its brakes, leading to runaway excitation and seizures, which is the root cause of Dravet syndrome. NAV1.2 is the backpropagator. It is largely responsible for sending the action potential backwards from the axon into the dendrites, a crucial signal for synaptic plasticity and learning. NAV1.6 is the trigger. Located at the nodes of Ranvier, it has a hair trigger sensitivity. It activates at very negative voltages, effectively deciding when a neuron fires. NAV1.7 is the amplifier. Located in peripheral sensory neurons, particularly pain sensors, it opens in response to small, slow depolarizations, like a receptor potential from a burn. Its opening boosts the voltage enough to trigger other channels. It is famous as the pain gene. Loss of NAV1.7 leads to complete pain insensitivity, while gain of function leads to man-on-fire syndrome. The second group consists of the TTX-resistant channels. These evolve distinct residues in the pore that make them immune to pufferfish toxin. NAV1.5 is the heartbeat. It drives the upstroke of the cardiac action potential. It differs from neuronal channels in its kinetics, having a slow activation profile suited for the rhythmic beat of the heart. NAV1.8 is the upstroke engine of pain. It is a workhorse that activates only at very depolarized voltages, but carries a massive amount of current. Once NAV1.7 amplifies a painful stimulus, NAV1.8 takes over to drive the actual spike upstroke. Finally, NAV1.9 provides the background hum. It has ultra-slow kinetics and acts as a volume knob, setting the resting excitability of pain neurons. While textbooks say sodium channels are for action potentials, biology begs to differ. Recent research has uncovered them hiding in unexpected places. One of the most alarming discoveries is the connection to cancer. Metastatic cancer cells often turn on VGSC genes, particularly neonatal splice variants of NAV1.5. Cancer cells use these channels to drive sodium influx, which powers the sodium-hydrogen exchanger to pump protons out of the cell. This creates an acidic microenvironment that digests the extracellular matrix, allowing the cancer cell to drill through tissue and metastasize. Even our immune system uses them. Macrophages, the white blood cells that eat pathogens, express NAV1.5 and 1.6 on the membranes of their internal lysosomes. When a macrophage eats a bacterium, the lysosome must acidify to kill it. The sodium channel allows sodium to escape the lysosome, balancing the charge as protons are pumped in. Without this sodium leak, the digestion fails. Another fascinating mechanism is the resurgent current. In some neurons, like cerebellar Purkinje cells, the channel reopens briefly during the falling phase of the spike. A peptide from the cytoplasmic tail of the beta-4 subunit jams into the open pore before the inactivation gate can close. When the membrane repolarizes, the peptide is kicked out, allowing a resurgent puff of sodium. This acts as a reset button, enabling ultra-fast firing rates. Because these channels are so critical, even a single amino acid change can be catastrophic. These diseases are called channelopathies. In epilepsy, Dravet syndrome is caused by a loss of function in NAV1.1. In pain disorders, a child with a null mutation in NAV1.7 feels no physical pain, walking on broken glass without noticing. Conversely, primary erythromelalgia involves a gain of function in NAV1.7, where the warmth of a blanket causes the hands and feet to feel like they are burning in lava. In the heart, long QT syndrome type 3 is caused by mutations in NAV1.5 that disrupt inactivation, allowing a tiny trickle of late sodium current that can trigger fatal arrhythmias. Humanity has been manipulating these channels for millennia. Tetrodotoxin from pufferfish plugs the outer pore, but batrachotoxin from poison dart frogs binds inside the pore and prevents it from closing, causing violent muscle contractions. In the clinic, local anesthetics like lidocaine act as use-dependent blockers. They are uncharged molecules that slip into the cell, become charged, and block the pore from the inside, binding best to open channels. This means they block hyperactive pain neurons more than quiet ones. The holy grail of pharmacology is selective painkillers. The problem with lidocaine is that it blocks everything. Recent breakthroughs like VX548 select target NAV1.8, offering pain relief without the addictive potential of opioids. As we look to the future, we are moving from blocking channels to editing them. For Dravet syndrome, gene therapy approaches like STK001 target the RNA to boost the production of functional Nav 1.1. Optical control is also on the horizon, with photo-switchable blockers that could allow for on-demand anesthesia with a specific wavelength of light. The voltage-gated sodium channel is more than just a pore. It is the fundamental unit of animal behavior. From the first twitch of a jellyfish to the complex arrhythmia of a human heart, these proteins tell the story of life in the language of electricity. As we master their atomic secrets, we gain the power not just to understand that story, but to rewrite it. Thanks for listening to G Fun Facts Online.